to introduce Professor Robin Hiesinger, who is a professor of neurobiology at the Free University Berlin in Germany. He also leads the Neurobiology Research Lab, which has lots of research, particularly on brain wiring, which I'm sure we're going to hear about today. So without further ado, I'm going to hand over to Robin, who is going to give us a very, very enlightening talk. Over to you.
Thank you so much for the introduction and really just having me here. So I do hope that you all can see my slides now, the little red laser pointer, and hear my voice. If not, just give me some shout out. And I'm, again, super glad to have that opportunity. As Keely just said, I'm a neurobiologist. I think that's not a very common thing to be for a speaker at a Computational Intelligence Society webinar. So it's an honor and an opportunity for me to reach out, as I have been for a number of years now, to a number of communities, including artificial life and neuromorphic computing, and now the computational intelligence community. Why am I doing this? Well, as a neurobiologist, I'm interested in how information is encoded in neural networks that lead to some kind of intelligent behavior. So is there such a thing then as neurobiology of artificial intelligent systems, is a question one may well ask. Now that you're here, I kind of hope that you feel it is an interesting question, but really what I kind of want to address or achieve is to answer the question, should one care about neurobiology at all? If one is an engineer, if one is interested in artificial intelligence, or all the different types of machine learning and computational intelligence approaches. The reason why I'm here is, of course, that there is something very obvious that we have in common, and that is the idea of neural networks. Neural networks, artificial neural networks, are currently the key approach to artificial intelligence. It's not always been like this, and we'll have to say a little bit about that to appreciate what neural networks bring to the table. For me as a biologist, of course, neural networks are everything there is. When we talk about brains, it's the substrate that does the computation, and it is what we are trying to understand in terms of how information gets into networks and allows them to do what they do. In artificial intelligence, I already introduced the brief notion that it's been a bit of a troubled history, rather. It's not always been neural networks. I brought you a picture here from the 1956 Dartmouth workshop. That's the famous occasion where AI got its name, actually, by John McCarthy. Other people here in this picture, some of you may recognize Claude Shannon, Marvin Minsky, Ray Solomonoff, and others. It's been an amazing workshop in the history of AI, not just for giving it its name, but also for two attendees. Herbert Simon and Al Newell actually brought to that workshop in 1956 the first AI program, really. Logic Theorist was a simple, logical, decision-making tree-based, and thereby symbol processing logic-based AI system, really. A rational thinking machine. It could actually validate and find even errors in some of the major mathematical theorems. And it was the program that was brought to the workshop. It has been, as we will discuss now for a few minutes, really at the forefront of AI for many, many decades, not neural networks. And yet, this is, of course, how we now, ever since computers use technology that was initially suggested by John von Neumann, this is how computers work to this day, but not how neural networks work, and not a brain. So when did neural networks actually enter the stage? About the same time, just 2 years later, Frank Rosenblatt very famously published the first neural network, the perceptron, that I'm sure most of you will be very familiar with. It's really been an interesting time. I mean, when this was published in 1958, like I think Science magazine had a cover article, "Human Brains Replaced," and New Yorker ran things. So it got very quickly quite famous, and it caused quite some backlash, not least by some of the attendees of the 1956 workshop that Frank Rosenblatt had not attended. It led to an interesting time for decades in AI research that was, as I said, dominated by symbol processing logic, but that had kind of neural network as a side stream. Here's Marvin Minsky, one of the really godfathers really for many decades of AI research. And maybe what happened to neural networks is best just depicted by telling you that Marvin Minsky, together with his then colleague Seymour Papert, actually published a book entitled Perceptrons a few years later, after perceptrons were initially promoted by Frank Rosenblatt and others, with the dedicated sole purpose of showing what perceptrons cannot do. That's quite rough. I mean, imagine hating a concept so much that you publish an entire book with the title of that concept just to show what they cannot do. Famously, maybe you know, XOR, for example, was one of the things that a single layer perceptron couldn't do, and others. Neither neural networks, then for sure, they were not at the forefront, but nor symbol processing logic actually met the early expectations of AI. And so what followed were what has very well publicized in a lot of beautiful literature on the history of AI, the so-called AI winters that really lasted on and off until like the early 2000s. And then in 2012, something very important happened. Some of you may remember the ImageNet competition that was first won by AlexNet, a neural network, a convolutional neural network, in 2012. And ever since, we've had basically a revolution of neural networks basically having taken over what we now call and are excited about as artificial intelligence. Deep neural networks is the name of the game, and big data to feed it. Now, topologically, we should note that deep neural networks are, by and large, multi-layer perceptrons. It's not like we've changed the type of network all them. Of course, there's now beautiful recurrent neural networks, and we can use reinforcement learning, and even convolutional neural networks that were originally used take some ideas from visual cortex in biology. But, and that's the key to any approach using artificial neural networks in AI to this day, these networks are all designed, built, and switched on to learn. There are differences to biology that we need to get to, but let me first then at least one more time emphasize what we really share with artificial and biological neural networks. And these roots grow deep. So this is then what I would say is the core or the origin of the neurobiology of artificial neural networks. First, 1943, the McCulloch-Pitts neuron. So Warren McCulloch and Walter Pitts famously wrote a paper that showed how neurons, computational neurons in a network, can do logical calculus. And any artificial intelligence based on artificial neural networks today fundamentally uses McCulloch-Pitts neurons as a component for computation. They're connected via some kind of synapses, we call them in biology. Synaptic connections is readily adopted by AI, and the updating rule for synaptic weights goes back to 1949. There is a book by psychologist Donald Hebb, who basically gave us the idea of things that contain information because there's coincidence, can be strengthened, whereas decorrelated information leads to weakening. And so this kind of idea, often dubbed cells that fire together, wire together, is, together with the McCulloch-Pitts neuron, really a foundational core of both biological and artificial neural networks. Now, some other things that we got very used to over the last years, like backpropagation, gradient descent, and others, are not so much found in the biological system. Nonetheless, current AI based on artificial neural networks is today closer to neurobiology than ever before in its history. So this is something to build on, I think. Something else that we have in common is a little bit more strange, maybe, and that is that none of us really know how information is exactly stored in these networks. So neurobiologists don't really know how, say, a 4-digit PIN of your credit card is stored in a neural network in your brain. We know the bit number of 4 digits, but we don't know how it is distributed, why these synaptic weights, why these 1,000 or 10,000 neurons that light up when you remember these 4 digits. We really don't quite know. Interestingly enough, now that artificial neural networks are so successful in AI, we're facing the same problem. It's often been dubbed like the alchemy of AI at this point. How is information actually represented in the network is not quite clear. And so this I find hugely interesting. It actually leads me to something that I just wrote down here so that I really don't forget to tell you. As far as I can tell, I think there is as much hope for neurobiologists like me to learn from AI researchers as there is for AI research to learn from neurobiology. Maybe if we understand how information is stored in artificial neural networks, we can actually look in biological brains and thereby learn from communities like, for example, the computational intelligence community. This all goes a lot into the direction of explainable AI, of course. In terms of machine learning approaches, this is a slide I actually saw at the computational intelligence Latin American chapter workshop in Bogota that I recently attended, and I was shown this, and I thought it was quite interesting that in terms of all the different machine learning approaches that can be used for AI, it's a beautiful inverse relationship of what works best, deep learning currently, and what is explained best, the classic symbol processing logic, with a number of other approaches in between. So clearly we have an explanation problem. We have an understanding problem. It works just like our brains do, but it's not quite clear how. We understand the math of the McCulloch-Pitts neuron. We understand the math of the updating rules. We don't understand how information is represented by and large in artificial or biological neural networks. Has some interesting effects. I mean, you may be aware, I'm sure you're aware of the Chemistry Nobel Prize this year, which was given one half to an amazing biochemist who did computational protein design, and the other half was actually for predicting protein structure. This is cool because 30 years ago when I was a student, this was a holy grail already. It was basically said then already. If anybody would ever manage to predict out of the 20 amino acids and in their sequence what structure the protein falls into, that should be a Nobel Prize. Well, here it is, solved by the DeepMind CEO and a research scientist at DeepMind for protein structure prediction. What's so interesting about this is that, of course, the DeepMind people are not protein biochemists. I mean, I don't even know how much they care. It's just a beautiful, beautiful data set. We just have a lot of amino acid sequences and we have a lot of solved structures. So if you feed that into a large enough network, it just predicts. It doesn't explain to us how it predicts. So in light of this, it's kind of curious that they wrote on the website of the Nobel Committee, they cracked the code for proteins' amazing structures. So I'm not sure there is a code. I don't know whether it's been cracked, how the amino acid sequence folds. Nobody has explained it to me yet, and the artificial neural network doesn't explain itself. It does predict, for sure. And I'll leave it at that. Not to take anything away from this amazing, very, very important step and award, but it highlights something important to us. And that is that when it comes to understanding where and how intelligence sits in the network, we have the same problem. Maybe we are on the same quest to explain artificial and biological intelligence. This is my segue to actually start to define a little bit, or try to define what we're talking about, artificial intelligence or any kind of intelligence. Let's talk about it. The definition of artificial intelligence that is most commonly used is, again, by Marvin Minsky, and it's been adopted by many others, and it runs something like this: the science of making machines do things that would require intelligence if done by man. Now, that's a curious definition because it uses the term intelligence to define some other intelligence. And so what is intelligence if done by men for some reason? Well, that's clearly biological intelligence. So we need to understand what that is, I guess, to use this definition up here. So what is biological intelligence? Not so clear. There are at least 15 competing ideas and definitions, and there's certainly no consensus amongst biologists. People mean very different things when they talk about biological intelligence, which could lead us to an interesting discussion about what we actually would mean with artificial general intelligence, but I'll not go there just yet. I looked up computational intelligence just because I got curious. So Wikipedia, as of today, defines computational intelligence. I don't look up neurobiology on Wikipedia anymore, so I don't blame you for not looking up computational intelligence on Wikipedia. But it's commonly defined or considered as a synonym of soft computing, but there is still no commonly accepted definition of computational intelligence, says Wikipedia. So maybe there's something else we have in common. Well, let me tell you something about the type of intelligence that we see in biology and how that may be of interest to the computational intelligence community. I'm a biologist, so here goes biology. Just lean back and just don't take it too serious when I tell you a little bit about what monarch butterflies do in North America. I love those. I actually lived here in Texas for almost 15 years, and they always pass by exactly when the passion flower bloom, and it's a beautiful migration route. That's what they're really famous for. So late in fall, when temperatures drop and millions and millions and millions of these butterflies are distributed all over northern North America, they all embark on a journey that is basically going to less than 1 square mile somewhere in the mountains in Mexico. And it's just absolutely remarkable that from all these different places, they find these few trees. And then they overwinter there. And as I said, then in spring is when we would see them in Texas. And what's curious about that observation is that these are the same butterflies that once flew south, but now that they eat and mate, they actually die. And there's a new generation of butterflies that picks up the trail north. They meet, they eat, and mate and die. And then there's another generation and sometimes another generation until you end up with a population of butterflies in North America, distributed all over the continent, that will embark when temperatures drop in late fall on a journey to a few trees in Mexico that were last visited by their great grandparents. Now, where is that information coming from? They are not communicating this to each other. They can't even learn it because it's so many generations later. It's really somehow an embodied network that was developed by a genome. Butterfly intelligence is grown. Somehow genomic information by a time and energy consuming process produces a butterfly. A butterfly's embodied neural network knows how to fly in 3D, how to navigate, recognize food sources, coordinate eating and sleeping, recognize mate with other butterflies, all prior to any learning. It's in the network. And now that is an interesting notion, because what does it mean the information is in the network? Well, we think it's in the topology, in the connections, in the synaptic weights of, here just a small section of an insect brain for which we can draw a wiring diagram. We like these wiring diagrams, of course, because in electrical circuits, that's what you do all the time. They're useful because you know how information flows. But the thing they clearly have in common is they're absolutely non-random. You need to put the components together just right to contain information before you switch it on. Now, that brings me back to the perceptron. This is a picture from the original 1958 picture. It's actually a visual perception system. That's how it was initially devised. So it has a retina, projection area. Here's the one layer where learning happens, the association area. And look what Frank Rosenblatt did. He just said, well, we're going to make random connections in and random connections out. Not really a butterfly. He actually went further. He actually said in the paper in 1958 something about biology. He said, at birth, the construction of the most important networks is largely random, subject to a minimum number of genetic constraints, said Frank Rosenblatt. And here it is. You can still visit it at Cornell University, the Mark I perceptron. randomly connected association layer. And this is how, and now it becomes kind of interesting, neural networks, multi-layer perceptrons of any kind of topology, however they're trained with big data, pre-trained, or reinforcement learning, they have, to this day, no genome, no developmental process. Artificial neural networks underlying all of AI today are designed and switched on to learn. The pre-training of, say, ChatGPT, of a transformer like GPT or DALL-E or many others, is still learning. It's not, there's no genome, there's no developmental process. It's just pre-training before you query it. So this is a big thing to leave out from the perspective of a neurobiologist. And this brings me to my first major point in asking a question about the relation of neurobiology and neural networks therein to AI and neural networks therein. In biology, babies, and for that matter, butterflies, are not born with random connections and switched on to learn. Quite to the contrary. We know that biological neural networks undergo a growth process that cannot even be clearly separated from learning. It's quite interesting. So if you look at the growth process of a neural network in biology, there's actually a time when it starts functioning. It starts learning, it starts firing. There's coincidence detection, Hebbian learning, synaptic updating, prior to any environmental input, prior to anything that could be learned. So learning becomes part of a genetically encoded growth process. And that's, of course, very different for the designed, switched on to learn types of artificial neural networks we have today. So to what extent do we want this in this? We don't know. And maybe we don't at all. Maybe this is, even from an engineering perspective, not the right thing to do or feasible. It depends on what kind of intelligence we're trying to produce. My task then now is to communicate with different communities what we think is the computational intelligence of the genome. So if this is such a big thing in biology, the famous 763 megabyte, that's actually all it is. We can save the human genome in 763 megabyte, somehow produces that thing, human brain. How does that work? Well, it's not a fair comparison. It's different types of information. Somehow these 763 megabyte don't compute to, whatever it would take to describe a brain. We don't even know how many bits we would need. Depends on the level of description of a brain. The genome contains information to grow a network, not information that describes a network. What should we compare? So we took a page of information theory and basically, so what is it that you need to compare if you're trying to understand the brain? For some reason, information theorists don't usually take the brain itself as an example for reasons that are kind of curious. They always use frogs. My best bet why there's like a series of books where this has been done. I guess they use frogs because it's more intuitive what happens when you put them into a mixer. So yes, some kind of frog soup. And that's information theory tells us what we should compare, because it's the same set of molecules could either be in a disordered state or in an ordered state. The frog really is nothing else but a stunning configuration of the molecules in the frog soup. And DNA seemingly plays a role in a time and energy consuming growth process, sort these molecules into that kind of stunning configuration. Now, is this enough? 763 megabyte. Let me ask an even more ridiculous question. How much can 0.000004 megabyte encode? How do I come up with that number? It's something I got very excited about when I first learned about it many years ago. Several of you, I'm sure, be very familiar with this. The artificial life community is gung-ho about this kind of stuff. Cellular automata. This is rule 110 of the one set of one-dimensional cellular automata described by Stephen Wolfram, the inventor of Mathematica. And you know how this works, I hope. Just very briefly, it's a deterministic set of rules that produces an iterative application, a certain pattern. So if I take the rule up here, it's 4 byte. of information, and I look up, say, this white square, and then I look up the rule, and it says make the one in the middle underneath white. And I look up white, black. Here's the rule, white, black, middle black, white, black, white. It's here, the one in the middle black, and so forth. So obviously this is deterministic. There is no random walk in this. And it produces a completely deterministic pattern. Here are the first 250 rows. The reason why I'm telling you about this right now is that there is a proof from Matthew Cook, still working together with Stephen Wolfram in the mid-90s already, that proved mathematically that this part here in the middle of the pattern will never repeat. It produces with infinite runtime any black-white, i.e. bit 1 and 0 pattern. And if you can produce any 1-0 bit pattern, you will produce a solution for any computational problem there is. This is the simplest known universal Turing machine. So there's a proof of Turing universality for rule 110. In other words, 4 bytes of information can produce infinite complexity. It's not the amount of information in the rules that determines the outcome's complexity. It's the runtime that leads to this. There's something even cooler about this from my kind of biological perspective about this. Part of that proof was not only Turing universality, it actually also included a proof that this is an undecidable problem. There's actually no way to predict, say, row 250 down here. If you want to know the pattern of this cellular automaton at any row, be it 100 or 1 million, there's only one way to find out. You need to let it grow, row by row by row. It's unpredictable, though it is deterministic. Strange. This is a good example for biology then. Yeah, this is a bit much to say. Obviously, biology is very different in many aspects. We think it's not deterministic, and especially we think that the rules change continuously as a function of gene products, and I will say a little bit more about this. However, this kind of abstract example of a one-dimensional cellular automaton highlights something very important to us. A very small amount of information in the set of rules can lead to, has no fundamental limit to how complex the structure can be that you can produce with it. So I'm happy to just opinionate here that there is no limit to how much information a genome can encode in neural networks prior to any learning, given enough time and energy for what I call algorithmic growth. Now, that brings us back to the genome. The genome with its 763 megabyte contains a lot of interesting stuff. The one thing it is not is a blueprint. A blueprint, and I'm showing you these wiring diagrams again, is a depiction of an endpoint. It's a depiction of the structure you're trying to produce. In the case of electrics, this is all the information there is, and it doesn't matter by and large in what order you put components together, as long as everything is in place before you flip the on switch. And that is just fundamentally not true for biological wiring diagrams like the one shown here on the left. The information for the outcome cannot be found anywhere. It has to unfold in a growth process. This thing here on the left has to grow. This one on the right does not. So what is the genome then if it's not a blueprint? The next best analogy is a recipe. Many famous biologists use that analogy, but it's not. The genome is not a recipe. Why not? Because a recipe contains a list of ingredients and a list of directions, what to do with the ingredients. And then you need somebody outside of the system to actually, put the chocolate, the butter, and the eggs together. They don't just jump together by themselves and create their own rules through interaction amongst the ingredients. But this is what biology does. In biology, there's nobody outside of the system who could read any directions, which are not written down in the genome. The directions come about through the interaction of gene products themselves, and they're continuously changing. So this is basically, in a way, biology 101, but it's hard to computationally realize and recognize the importance of this. So biology 101 is what the genome really encodes, of course, is, proteins. I mean, that's actually not entirely true. We now know there's actually in the human genome more non-coding genes now than there are coding genes. starting this year, and the tally is still ongoing. So the genome does more than just coding for proteins, but for simplicity, even if it would, the proteins themselves start to interact with each other. They form higher order assemblies, including proteins that go back to the DNA and change what is going to be read next. This is something of a feedback system that in biology we call transcription factor cascades. We have continuously changing states in time. Say you have a gene that produces a protein, then that protein goes back to the DNA and turns on the next gene, which produces a protein, which goes back to the DNA and turns on the next gene, and so forth. Now, this is something you can already not read in the genome. These are rules that come about through the gene products themselves. So the genome encodes something in feedback with its own products. Now, what does that sound like a lot? Well, self-organization. So the genomes and the rules are not external lookup tables of what to do with or how to sort gene products to create the states of the cell. The gene products and their higher order assemblies create their own rules in interaction with themselves and the genome that encodes them. Pretty tough stuff. I will say a little bit more about self-organization in a minute, but before I get to that, I just feel like, now is the time to relax a bit and just look at some of the biology that led us to do this. The computational intelligence of genomic encoding is clearly an evolutionary programming of genomes that has identified amazing solutions to complicated problems of embodied intelligent systems. But the genome does not encode the outcomes. It encodes self-organizing processes that produce outcomes that are sometimes super precise, but at other times more adaptive and also robustly variable. So how that works is something that my lab actually studies. So this is where neurobiologists like our laboratory and a larger consortium come in, and we're looking at model systems, as we call them. Here's our favorite one. This is like, kind of a fly. This is nonsense, actually. Flies don't really look like this, but more or less. Here's a bit of a more schematic depiction of a fly head that I'm going to take apart for you, just to give you an idea of the type of network topology that we're confronted with in biology that we're trying to understand. What I've taken out here is an eye from that fly, and I can take out the entire brain. So yes, flies do have a brain, actually quite complicated, 100, 200,000 neurons. The eye consists of many, many single eyes that I hope you can recognize here in this movie. And each one of those single eyes, we're zooming in, is connected via a set of cables to the brain. These are axons. Axonal cables are the output parts of neurons. And what's curious, if you look in the brain here, these cables all, I hope you can see this, are actually plugged into separate units in the brain. If you follow the cables from the same unit in the brain, they are plugged in to separate different eyes. Strange. This is a very complicated wiring diagram. It has a name. It's called neural superposition. Actually, the early cyberneticists in the 60s, there was a Max Planck Institute in Germany here, they studied this a lot. They're really hoping that if we understand how biology creates such a complicated wiring diagram, maybe we can learn something about what they back then called machine learning. But really what it does is very simple. It looks complicated, but really what it does is it takes input lines, in this case from the fly eye, that are in different eyes, but look at the same point in space. And at the level of the brain, you have an ensemble, a synaptic unit that literally just looks at a single point in space. So it's a visual map. And we have that as well. So humans have, a visual cortex, for example, is a depiction as synaptic units of neighboring points in space mapped as neighboring synaptic units in the brain. Now, in terms of genomically encoded unfolding of information, it is a bit of a problem because these cables initially grow down to the brain as one, but then each one of these cables has to go somewhere else. How does that work? Well, we know a little bit about just the anatomy of this and the dynamics of this. I have a little schematic here for you, just again to give you a bit of an appreciation of what happens in a neural network in biology just to get the wiring right. Here, the 6 cables are coming from a single eye. They actually all look at different points in space, so they need to be rewired. They rewire in this sorting plane that we're now taking a top view of. And this rewiring is schematized here as like the orthogonal to the initial axonal growth of these cables. In biology, these are so-called growth cones of axons. If we now follow a single one of them, say this red one here that projected to this green blob, the green blobs are the different target regions, then we find that all the others from the same went to different target regions, but this particular target region here is innervated by 6 different ones, yet from 6 other bundles. These are all the photoreceptor axons receiving the same visual input, looking at the same point in space. So what you see here is a schematic, is literally a developmental process that has to happen in the fly brain just to wire up everything right prior to any learning. This happens during development in this sorting plane. These fronts of these growth cones then grow underneath. So basically think about the top here as input, the middle as computation, the bottom as output. And you make columns, just like our cortical columns in the human brain. And these columns here are literally synaptic units that are wired correctly. It's cool because this sorting plane is actually something that only lives for 15 hours and then goes away. And we can look at how this happens live. So what you see here now it gets really biological. This is actually two-photon microscopy is a special type of intravital non-invasive imaging technique that allows us to look inside the brain of a fly without affecting its development non-invasively, and the fly survives this while rewiring happens. I hope you can see my pointer here on the left side. This will be one developing eye, and we're labeling a few individual cells and the cables growing into the brain. And this region here in the middle, this is the sorting plane. This is where the rewiring happens, as seen live now over 15 hours. Now, this is the type of, we call it 4-dimensional data sets that we can obtain from developing fly brains. We can then rotate them, we can play them forward and backward. This is now the view from outside the eye. You can see the beauty of the many different single eyes. You can see that we have stochastically labeled only very few cells. That's important, so we can see a tree inside that forest. We can segment out with digital image analysis only the ends of the cables, these growth cones, and then we can look at their dance. Voila, what you see here is the rewiring of network topology inside the brain of a fly. We can look at the outcome. We can then see, oh, these look similar, these look similar. We can give them certain colors. We can follow the cables back to the cell body positions in the eye that allows us to determine precisely what the individual cell types are. And then we can do basically the final type of movie, which is the dance backwards from the outcome, where we know what these subtypes are. It doesn't matter what name they have. And here you see basically false colored rewind to an earlier time point when you would not have been able to predict what these cells are. So this is what happens live. Input of cables, resorting, a type of temporal and spatial computation to change the network topology in a genetically encoded program prior to any learning. The structure vanishes, literally. There's no trace of it anymore later. And you end up with a perfect wiring diagram that is important for the adult neural network to do embodied neural network computation. How does this work? I'm not going to tell you all about a recent paper in which we kind of figured out to a large extent how this works. I just want to give you the essence of something surprising that we found. The surprising thing is that, I guess as electrical engineers, if you want to put something together, right? One of the key things you want to know, who's input, who's output. What's my target? What is the socket that I put my plug in? And what we basically checked in biology is, well, what's the role of the targets? So all these green blobs here. And the answer is you don't need them. It's a pure self-organizing system. How did we find that out? Here again, I'm showing you a 2-photon imaging of a living fly during development. We're coming from the eye. We're segmenting out now in this glow color map just one of these cables times 800. That's the number of pixels the fly has. And here in green, all the so-called postsynaptic cells. So the ones that the The glow-colored map ones, the beige ones here are connected to. And again, this is live data. So we see inside the fly's brain how, 800 times a certain cable needs to grow to the right. This is over the next 10 hours. After 10 hours, there's an important time point that for biological reason we call 35% of development. You see these vectors have become longer and they're at the target region. If we now start killing all the targets or even just some of the targets, here's an experiment where we sparsely killed just some here and none here, but then you see the green cells start to lose organization. The cables that we're interested in that do the neural superposition, rewiring, don't care. So over the next 10 hours, you have a deteriorating field of targets. So the sockets your plug's supposed to go into, but the actual plugs, the actual cables are perfectly fine. You would not be able to tell from just looking at them that we've completely genetically messed with their targets. How does it then work? Well, all there is are 4,800 searching cables that have nothing but 4,800 searching cables as guidance. By 4,800, because these 6 subtypes exist in every single one eye, and there's 800 of them. So the fly is pretty crappy spatial resolution. It's 800 pixels, but very good temporal resolution. So this is all there is. How does this guy, for example, go from here to here? Here we have a depiction of that time point where you see many of these protrusions. You see these little, very biologically looking, like fiber-like things. These are so-called filopodia, membrane protrusions that are very dynamic. Here is one vector that basically tries to grow the red dot away from the blue dot to this target region. The filopodia don't sense the targets. They're actually, if you make a whole density map of membranes, they're just going wherever they're not prevented from going to. The entire system is true self-organization in the sense that all you need is 4,800 of these searching cables. They will create this density map, and within this density map, if only sent with the right polarization on the initial way, will create a growth vector that is basically the integral of all these small filopodia. Can we show that this really works? Well, here is a full mathematical simulation of the process. This is the visual map as it develops, the sorting plane that only exists for 10 to 15 hours. And now we simulate basically these filopodial placements. We simulate the average weighted vector that allows them to grow up to the time point when they are already at the right target region. And the whole thing works without any outside information. So this is what this looks like. And this is kind of why we call this self-organization. So let's just have one more word on self-organization, because it's also, especially in biology, a bit of an overused term. We use a very strict definition here. We're basically going back to Ilya Prigogin's idea of dissipative organizing systems. Here's a famous example of self-organization, maybe the poster child. You know, when you heat a certain viscous liquid on a hot plate, there are states dependent on the viscosity and the heat distribution that has to come from one side, where you get these convection rolls. And this is important because it's an organizational feature that comes about through the components, the microstates of the system, interacting with themselves. There is nobody outside the system that tells the molecules in the liquid to start to make these particular rolls with these particular diameters, with this particular speed. So this is true self-organization because it's the agents of the system that are being sorted are the ones that through their interactions create the rules of organization. And that brings us back to the comparison to wiring diagrams, blueprints that are outside information. So that's what the genome can't be. Recipes that require somebody sitting from the outside to put the eggs and the chocolate together. And that's not what we're seeing. What we're seeing is the microstates of the system as part of a genetically encoded process through their own interactions, creating the rules of the sorting, the rules that create network topology. So in sum, what this to us biologists means is that there is a very important information encoding process that we are studying, that my lab, for example, is studying, that is very closely related. In fact, it kind of transforms into learning, including like Hebbian updating rules of synaptic weights, but it's not separate from it. And it requires a genome-driven developmental process to come about and encode information in a network prior to any learning. So information encoding through self-organization requires no director outside the system. The directions, i.e. the rules, grow. The agents do not know what they're doing and yet produce a topology that is evolutionarily programmed. And this is actually a scale-free concept. It's a concept that applies to our butterfly. The butterfly, as an agent, does not actually know the route of the migration that I have shown to you at any point either. The route is actually not saved anywhere in the network. There is no Google Maps for monarch butterflies in the US. The route grows literally in time through the interactions of the butterfly with the height in which they are flying, because there are certain streams that help them get to the right place, the magnetic field, day-night cycle, and landmarks. So the agents will create very robust, should we call it topology of a migration route, without themselves receiving any rules from the outside. This is the idea of information unfolding, and this is the idea of computational intelligence of neural networks when we look at it from the perspective of biology. We have feedback systems. We have a genome that produces molecules that do molecular computation. Molecules, as we have seen, go back to the genome and change what molecules next will do a different computation. Molecules form subcellular structures, a new organization level that itself does computation. The Philopodia I've shown to you are agents that are not controlled by a single molecule, yet feed back to molecular computation, feed back to the genome. So does cellular computation and, of course, network computation. And these feedbacks are not just one level. We have cellular feedback to the molecular level and molecular changes to cellular computation. We have feedback between subcellular and network levels and network and molecular computation. All of this is part of information encoding through algorithmic growth in biological neural networks. How does it compare to artificial neural networks? Well, we're currently here. Artificial neural networks do leave out, for good or for bad, the genome, molecular computation, and subcellular computation. We have exclusively cellular and network computation in feedback, especially, reinforcement learning is the poster child of this type of feedback. And we all know that once you introduce feedback into a system, the sky's the limit. So I cannot conclude that this is a bad thing. We don't know. But I can tell you that in biology, this would not get very far for the intelligence of a butterfly. And this up here is clearly needed for human intelligence to wire up information in babies' brains prior to any learning. It's a big thing to leave out. It's an interesting thing to discuss. And with that, I'm thanking people in my lab. I'm thanking very much the IEEE Computational Intelligence Society for allowing me to reach out. I'm grateful to Princeton University Press. Some of the ideas I presented to you today are published in the book, The Self-Assembling Brain. And I thank you very much for your attention, and I'm happy to take questions.
Thank you so much for that amazing presentation. I don't even know where to actually start, but we have got a question. I'm going to read the question out for the benefit of everybody. It's quite a long one, but you should be able to see it in the chat under panelists as well, but nobody else will be able to see it, so I will just read it out. And it's from Chris. And he says, it seems that today's artificial neural networks are not completely random, since they are not just fully connected networks, but are significantly constrained. For example, the convolutional structure of CNNs, the residual structure of ResNet, and the attention structure found in today's foundation models. And the question is, what is the role of these structural constraints relative to the initial neural network weights, which are random in today's neural network approaches? And last bit, is there a comparable distinction in biological neural processing? Over to
Thank
you.
you. Thank you so much for that question. I mean, you can guess the answer already, right? I mean, it's a beautiful question. And the answer is, of course, yes, it's hugely important to have these non-random topologies also in artificial neural networks. And yes, of course, we know those from biology. Like convolutional neural networks are maybe the ImageNet 2012 winning AlexNet was a convolutional neural network already, and there's a good reason for their parallelization and how they can do particularly well with image recognition tasks. There have been famous proposals quite recently. I mean, Geoff Hinton has always said that we need to come closer to the brain, and of course, some of you may have read Jeff Hawkins stuff on how we can maybe make artificial neural networks more using columns as they're observed in our cortical columns. I introduced to you a little bit visual cortical columns. So the answer is there is a lot that is being very much appreciated in the artificial neural network community in terms of what we can learn from topology of outcomes of growth processes from biological neural networks. And you see where I'm going with this now. What's not included is how you got there. Yes, we can improve both topologically by doing columnar structures and non-random connectivity to some extent, as we can improve and have dramatically improved, as DeepMind has shown us, with reinforcement learning, which is of course something also quite biological. The idea that you learn from outputs of previous iterations of computation. But this is literally the very, very surface of what we see finished brains do. What's not included in any artificial neural network is a genome that actually drives this. And that information in feedback with its own products does a lot of computation in mine and your neurons right now. And there are examples of even just dendritic computation of a single cell. You need like 100,000 neural network, nodes neural network just to simulate that. So that's how much computation, even at a superficial level, a single neuron in the brain can do. And so it's an interesting question to what extent we can do what I just described, maybe simulate all the more complicated molecular subcellular jazz by just standard artificial neural networks. Energy is going to become very quickly a big problem, as we all know. But more conceptually also, the way the feedback works can't easily be simulated. If you do neural networks for each component function, the way they interact is not the same as the computation that agents do in a biological brain. And I need to stop answering because otherwise I keep on talking.
No, I think it's just a fascinating topic and there really isn't enough time, but there's two other questions that have come in, which you should be able to see. So just for everybody else, from Stefan, are you arguing that artificial neural networks will greatly evolve by using feedback loops in a modified pre-training to create a self-organizing phase where the network grows prior to learning through exposure?
Wow. Yeah, I'm not arguing this just because I don't know. I mean, I'm just putting what we study in biology out there and kind of say, look, we know that there's a lot of computation happening in these feedback loops between molecular, subcellular, cellular, and network levels. To what extent this can be used or should be used in artificial neural networks, I mean, quite frankly, it's your job. I can't program these things. I study them in biology and I already don't understand those. I hope and I think that there are approaches that one could take. So to be a little bit more hopeful maybe or positive on this, we did collaborate a bit with some artificial life scientists who actually do, as many of you will know, some neural network training, not through learning, but through evolution. You can have, like, say, a gene matrix where each, in direct encoding, each value, each gene, if you will, is one synaptic weight, which is not how biology works, but at least it's something. You can do indirect encoding like HyperNet and HyperNet, and some of you may know these kind of techniques, and basically train neural networks like that. So no training other than evolution running through, filling a synaptic weight matrix, running a recursive neural network, having some agent go through a maze, and then go back to the genome. Super, super, super tedious, even like orders of magnitude more computational power needed to simulate. They do work, but they don't work better than the learning mechanisms that we see currently being successful in AI. I'm curious though, because I told you a bit of that story of neural networks, right? Until 2010, if you asked, I just say, please object if you think I'm wrong on this, right? But anybody I talk to in neuromorphic computing, A-Life, or recently at the computational intelligence, everybody is literally saying, we were so surprised when neural nets were suddenly so successful, because we saw them, we worked on them, we thought they were great, but we never knew and we never expected that suddenly, like in 2012, they can do everything. And it was not the topology that changed. It was literally just the computational power needed to simulate them and the amount of big data to feed them. So maybe what I'm just describing, training neural networks based on this much more cumbersome evolutionary process, has not hit that, is still below that glass ceiling. I really don't know. I do think it's worth talking about. I think it's worth recognizing what type of information encodings are out there and to what extent, for different purposes, they may be applicable.
Okay, wow. Okay. And the next one, I'm sorry. How would the neurons differ from a perceptron approach? Is it a linear sum with logistic?
How would what neurons differ from a perceptron? The neurons that, like, if you take what I say as a proposal, you mean, if you think of, what does Robin want? Does he want us to now, simulate not just McCulloch-Pitts neurons with Hebbian synaptic weight updating, but he wants us to simulate every single molecule? Then I agree. I think that's hopeless. That's, it's also not how biology works really, right? Because this idea of simulating all of this on a von Neumann architecture computer, it blows up very, very quickly, as you all know. Some famous companies have just bought, like, nuclear power plant companies to just build those, and some other people are looking for nuclear fusion to solve this problem. But there are other solutions. Biology has found other solutions. If you let agents interact with each other to go through this type of growth process, you can encode information, but you have to program it via an evolutionary mechanism. Genetic algorithms are doing a bit stuff like this, right? So how would the neuron differ from a perceptron? I would do baby steps. I would do baby steps of introducing, just as it's being done, right? Topology, maybe information encoding, where it's not just a McCulloch-Pitts type neuron, but it's also a neuron that has, maybe co-transmission of an excitatory and inhibitory transmission, which is the standard thing we find in the brain. You don't only have synaptic weight that is more or less, you actually have opposing directions of exciting or inhibiting. You can do this with neuromodulators. There's a lot of proposals one could test. There's clearly lots of questions on feasibility.
Okay, there's just one last point because I know we're on time, but I think it's really good point from Paul as well, and I guess really opens up the whole topic further. AI is seen to be useful, but is it really only a machine learning subsystem? And real artificial intelligence will need another method. I think it's a really great point to end on. Please, can you reflect on that, Robin?
Well, I just, you said it. It's I personally just want to be very careful. I just want to learn. I'm very grateful for this opportunity to discuss. It's super interesting to to learn from different communities about how they think about what is surprisingly similar problem. I mean, we are in biology trying to understand how information is encoded in neural networks. And it's interesting how then many people, for example, in the computational intelligence community, are surprised how little neurobiologists actually understand about their brains. And so I think the point is very well taken. It has been made by a number of people. There's, of course, also always this perspective of, well, we're very excited about what works. We don't actually care about how it works, as long as it predicts what, color of socks we're going to sell you next. And so we don't even want to know how information is represented in the network, as long as, we can sell you stuff. Most AI that is being discussed in our field is just using it as a tool. The big discussion about what types of intelligence we're producing is a super interesting and not very well-based discussion, because we're by and large we don't know, and the protagonists in this discussion by and large don't know what they're talking about. What is intelligence? I gave you the butterfly. I can give you many other amazing examples of animal intelligence, what octopus do, for example. And human intelligence is not great on any single task. It's outcompeted on everything. You can, there's a network that plays better chess, that does better image recognition, that does better cocktail party problem solving, anything. But that's not human intelligence. Human intelligence is not on a scale. Human intelligence is doing 100,000 things in a mediocre way, but at the same time in feedback with each other. How much I love my wife should not, but probably occasionally depends on how hungry I am and when I have ate last Sunday. I mean, I hope she's not listening to it. The metabolism of neurons affects how they function, and there's feedback between those. This is part of human intelligence. It's not just playing chess. So in light of this, what is artificial general intelligence? I have no idea what this is even supposed to mean. There is no such thing as general intelligence as far as I'm concerned.
That is absolutely fantastic. And I was going to end there, but there is a really good question. I think this is definitely for you. So, do you recommend any book or piece of literature to help understand this concept better? And anything from an AI researcher perspective on what they would understand? Because I think that is the big question from today. You've raised so many issues and future directions of research. What would you recommend?
I mean, I have to, of course, recommend the self-assembling brain. I mean, that's, I wrote it for that purpose. But it is written from the perspective of a biologist. Now, from the perspective of an engineer, I personally did enjoy Jeff Hawkins' first book on intelligence very much, more than the second. But they're both super interesting. And, he's an engineer who describes how when he, in the 80s, actually wanted to learn from biology, was discouraged from doing so. Wrote about that, both from a historical as well as like conceptual and computation perspective. Other than that, I should probably be giving you like 5 other books right now, but I can't like come up with them at this
It's
point.
okay, Robin.
Geoff Hinton, like anything on YouTube you find, there's like a beautiful talk he gave, I think at MIT in 2014, for example. Has been very, not only upfront recently about his concerns, but also about how whatever artificial neural networks do has to learn from biology. It famously argued a lot in recent years against backpropagation. And I think I leave it at that.
Okay. Well, I'd like to thank Robin very, very much for probably was amazing insights. I've learned so much. And again, I guess I was one of those AI crowds that grew up in the neural network era a long time ago. So thank you very, very much for that. I think there's so many questions here. So we may have you back again. Okay.
Please don't hesitate to shoot me an email. I'm not on social media. I don't like that stuff, but I do respond to emails really well.
Okay, that is great. So I'd like to thank everybody for joining this webinar. This recording will also be available as well. I'm sure there's so much in it, we'll watch it back. And thank you once again to Robin. I'd also like to thank Tom and Joellen as well from IEEE, who have also made this happen. So thank you, everybody. And wherever you are, good morning, good night, and good evening.
Thank you, everybody, so much. It's an honor.